second part of chapter 4, we're going to focus on the difference between gram-positive and gram-negative cells, and then talk a little bit more about some of those internal structures of prokaryotic and bacterial cells. So first of all, just one of the most important tools for identifying bacteria is the gram stain, and because it identifies the difference between those two major groups. And the difference between them is based on that cell envelope, so their cell wall and what's outside of that cell membrane. The reason it's called the gram stain is because it was developed by Hans Christian Gram, so he got to use his name in this stain. So our two different types of bacteria, we have gram-positive and gram-negative. And the difference comes in that cell wall. So they both have a cell wall outside of that membrane. The gram-positives have a very thick cell wall. And that cell wall in both of them is made up of a carbohydrate called peptidoglycan. And then that's on the outside of that inner cell membrane. So they just have the cell membrane and a really thick cell wall. Gram-negative cells also have a cell wall, but it's much thinner. They have that cell membrane, but then they have that additional outer membrane. So they have the normal cell membrane, a little bit of cell wall with that peptidoglycan, and then this outer membrane. And this can be seen here, and it also shows, so this is just a nice, especially if you're a visual, that can kind of help you hopefully remember the difference between these two. So again, they both have this cell membrane made up of these phospholipid bilayers, allows them to control what's coming in and out. But with a gram-positive, on the outside of that cell membrane is this really thick layer of peptidoglycan that makes up their cell wall. They also have a compound called teichoic acid. We'll talk more about coming up here in just a minute. So that's the gram-positive. Then the gram-negative, they still have that cell membrane. They have a cell wall with that peptidoglycan, but it's much, much thinner. And then outside of that, they have this additional membrane called the outer membrane. Now, it doesn't keep as much from coming in. It's much more porous. You even see these little porins here that let things come in and out. But again, it's just another layer of protection. So whenever we're treating infections, it's important to know the difference between whether it's gram-positive or gram-negative. It's one of the most important tools to identify a bacteria. So just looking at these a bit more, so some of the characteristics of that cell wall, that also determines the shape. So whether it's coccus, bacillus, curved, that's determined by this cell wall. Again, it's a very, rigid layer. It provides that strong structural support because, again, these are mainly single-celled organisms. So they don't have skin, they don't have bark, they don't have anything like that to protect them, especially from either collapsing or bursting due to changes in osmotic pressure, which is based on the fluid there around. If there's a lot of solute in the solution there around, that can cause water to go in and fill the cell, and it would bust if it wasn't for the cell wall, or it could lose its shape if water's coming out. So again, the cell wall helps protect it. And again, both in the gram-positive and gram-negative, so all bacterial cell walls are made up of this peptidoglycan. It's a unique macromolecule. It's not something we have. So a nice target for antibiotics, because if we can inhibit this compound, we don't make it. So there are less side effects. But again, it's made up of a carbohydrate with some short peptide or protein fragments in it. So it's strong, as we've mentioned, but it is also flexible. So there is some moving and bending that can occur to allow all the cell functions, again, things coming in and out of the cell. It's rigid but flexible, if that makes sense. And this is just looking a little bit at how the peptidoglycan, so again, it's made up of these alternating sugars called glycans, hence the name, but it also has peptides. So peptides and sugar essentially is what we have here. But it alternates and then crosses back and forth to form this cell wall. So whether it's thin or thick, that general structure is the same. So looking a little bit more at the gram-positive cell wall, again, very thick peptidoglycan, just to kind of put it in terms, 20 to 80 nanometers. You're not expected to know the thickness, but just to give you an idea. And then again, it has this teichoic and lipoteichoic acid in it. that helps maintain that cell wall, just keeps it intact, keeps it from kind of falling apart. And it's enlarges, gets bigger during that cell division, and that acidic charge, the acid of it charges the membrane, so it makes it positively charged. The gram-negative cell wall, though, in comparison, still has peptidoglycan, but only about 1 to 3 nanometers thick. So again, at least 20, almost up to 100-fold shorter or thinner than the gram-positive. Again, don't need to know those numbers, but just to put it in perspective. So it's still somewhat rigid, but definitely not as much as that thick gram-positive cell wall. So it provides greater flexibility and an advantage, but it is it is more sensitive, more likely to go through lysis or rupturing when it changes that osmotic pressure. And then another thing that these gram-negative cells have is called LPS, or lipopolysaccharide. This by itself, if you would inject an animal or a human with this, automatically causes fever and an immune response. So this is only found in the gram-negative. So teichoic acid in gram-positive, lipopolysaccharide, or LPS, in the gram-negative. So those outer layers have these different compounds in them that make them unique. All right, now looking at some of the internal structures, again, the cytoplasm, that's just kind of a gel-like solution inside. That's where most of the activity is going to happen, as far as if they go through photosynthesis or getting and using energy through fermentation or respiration, cell division, all that. All the activity is really happening in the cytoplasm because they don't have organelles. So it's about 70 to 80% water, and then it has other solutes as well, like sugars and amino acids, different salts, different enzymes, that type of thing, as well as containing some of the other things we're about to talk about. So like ribosomes, different cytoskeleton parts as well. Again, the plasmids we mentioned before, but again, these are non-essential pieces of DNA. So extra DNA that's separate from the main chromosome that has all the required information, still double-stranded, circular, just like prokaryotic DNA is. But whenever the cell divides, it's duplicated and passed on to the offspring. So not only do they get the main chromosome, they also get all these plasmids as well. So the offspring get any advantages. And they provide some various protective traits, especially, again, like antibiotic resistance. But as far as using bacteria in research, genetic engineering uses a lot of these plasmids to add different genes or different traits to bacteria. So for example, there are bacteria that produce insulin because the insulin genes placed on a plasmid inserted into some bacteria, and bacteria start making insulin for us. All right, then our ribosomes, all cells actually have ribosomes, and these are made up of RNA and protein, and they're found throughout the cytoplasm, usually in some chains in prokaryotic cells. Now the difference, though, is the bacterial ribosomes are a little bit smaller, so 70S, and that's a Svedberg unit, which is just like a relative size, not a precise like nanometer or meter, inches. But again, the main thing I'm highlighting here is that bacterial ribosomes are a little smaller than eukaryotic. So they both have them. They're used to make protein that the cell needs using instructions from DNA. But the bacterial ones are slightly smaller. So again, it provides that nice target for antibiotics. And then those endospores, again, their advantage is it's a dormant state, so they can withstand really hostile conditions that facilitate their survival. So some of them can live for years in soil. So that's why whenever you have a wound, it's important to make sure a tetanus shot is recent, that it's cleaned and irrigated really well to get out any soil. And again, the main issue with the soil is that endospores can be present there. They're dormant, but once they get inside of a human and the temperature's all nice and they have all kind of nutrients, then they become active. So they have kind of a two-phase life cycle. The vegetative, that's when it's metabolically active, and then the endospore is that resting state. So again, it's not going through replication. It's not doing like respiration or fermentation. It's just a dormant state. And that process of going from the active vegetative form to the endospore is called sporulation. So it's the process of forming a spore. It's not a reproductive method. It's just a way to become dormant when things are hostile. Now, endospores are the most resistant. So this is, whenever we're talking about sterilization, this is what the key is to remove endospores because they're resistant to heating. So boiling something does not destroy endospores. They're resistant to being dried out, resistant to temperature changes in the opposite direction. So even whenever they're frozen, even radiation that's usually used in a lot of different types of sterilization or just disinfecting and various chemicals. So again, in order for something to be sterile, the main thing that we're looking for is the removal and destruction of these endospores because they are the most resistant state. Now, fortunately, not very many organisms can form these. So just Bacillus and Clostridium, so like tetanus, anthrax, they're examples that can form these endospores. And so just to kind of highlight some of these, so again, anthrax, Bacillus anthracis, that causes anthrax. Many years ago at this point, there was actually bioterrorism where these spores of anthrax were placed on envelopes as a way of again, trying to harm other people because, again, they're dormant in the male, then they get on skin, they get in the body, and they start producing the disease. Again, talking about tetanus and making sure wounds are irrigated correctly, Clostridium tetani is one that can cause tetanus. It's a spore former, gas gangrene, and also with canning and food, Clostridium botulinum that causes botulism is one of these spore formers. And then finally, just some of the medical significance of these. Again, they're constant intruders where we're worried about sterility and cleanliness. Again, most resistant to ordinary cleaning products like boiling or soaps. So they're the ones that are usually contaminating different cultures and media. And again, especially in the medical field where we have different wound infections that lead to worse problems. So also finally, just getting rid of those is important in canning and food industry to remove those so that they don't cause foodborne illnesses.